Good afternoon or good morning. So my name is Boonen. I work at the University of Antwerp, but I am also co-founder of ImmuneSpec, which is an immunopeptidomics company that was founded this year. So I think the previous talk was very interesting and also made it already clear that there are a lot of developments in the field of immunopeptidomics, and that these technological developments make that immunopeptidomics is evolving at a very fast pace. So yeah, we can now do a lot of things that 10 years ago were completely impossible. So these developments have an impact not only on precision medicine and immunotherapy, as Professor Purcell showed, but also on vaccine development. And the thing that we mostly are interested in our company, or is a service that we provide, is actually the immunogenicity testing. So together with my colleagues here and Elise, we started in the immunopeptidomics field, let's say 5 years ago at Antwerp University, and also at the Flemish Institute for Technological Development. So after a series of contacts and collaboration requests from biotech, we decided to create a spin-off ImmuneSpec, and that's now located in the Antwerp region. So we work on applications of immunopeptidomics in medicine, but our main focus is, like I said, the immunogenicity testing on biotherapeutics. So biotherapeutics or biologicals are drug therapy products which are active substances that are extracted or produced from a biological source. So for these biotherapeutics, immunogenicity is so an immunogenic response is of course the opposite of what you want. Oh, excuse me. So biotherapeutics, they are very useful for treating some difficult conditions, and around 350 million patients a year benefit from these biotherapeutics. So they can be used to treat diseases such as cancer and diabetes. So we all know the monoclonal antibodies that act against cancer drivers like cetuximab, which is, for example, an EGFR inhibitor, but you can also have antibodies against other proteins like infliximab, which is a TNF-alpha inhibitor, and it's used in autoimmune disease. So biotherapeutics, they are not only limited to antibodies, and of course the development in computational biology and artificial intelligence make that the design of new protein-based therapeutics is booming. So some of these therapeutic proteins or also some parts of these proteins, they are not expressed endogenously. So they can also have like modifications, or they can aggregate, which makes that they will actually be perceived as non-self to the patient, and then they can invoke an immune response. The key process for invoking this immune response is, of course, the presentation of the peptides by antigen presenting cells, and this happens at the MHC2 complex. So these peptides presented by the complexes on the surface, they can be perceived as non-self by T cells, which after they get co-stimulatory signals and secrete cytokines, they can actually stimulate T cells to proliferate, but they can also lead to the production of antibodies by B cells. So of course, the last thing what you want is that an activated B cell makes antibodies against your biotherapeutic, of course. So in the case of vaccines, the thing that you want is actually this immune response. But of course, for the biotherapeutics, that means that there will be a clearance of your therapeutic protein, and you can also have side effects, since these anti-drug antibodies, they can, for example, they can target endogenous proteins with cross-reactivity, and they can have severe detrimental effects. So the formation of anti-drug antibodies can thus limit the efficacy of therapy in the case of repeated administration and it may potentially induce severe unwanted immunogenic responses. So a challenge in biotherapy development or biotherapeutic development is therefore to de-risk candidate drugs and to develop biotherapeutics which have a minimal immunogenic potential. The process from antigen presentation to actual anti-drug antibody production, it cannot yet be tested in vitro. So in vitro assays like T-cell assays, like measuring T-cell proliferation or T-cell activation, or also B-cell activation assays and cytokine release assays, they can give you information on the immunogenic potential of a biotherapeutic in the target population. However, these methods, they cannot pinpoint which part of your therapeutic protein is causing the immunogenic effect. So MAPS assays, so MAPS means MHC-associated peptide proteomics, which is actually a synonym for immunopeptidomics that's often used in the field of immunogenicity. That enables you to precisely identify the region of a protein that may evoke this immune response. So key applications include, of course, the testing of biotherapeutics, but you can also, for example, stimulate cells with a pathogenic protein and then look at which part of the protein will be presented, and then you can also, of course, enhance vaccines. So for the identification of possible immunogenic regions of a biotherapeutic protein during preclinical phases, so antigen-presenting cells are loaded in vitro with the target biotherapeutic, and then they are matured. So these antigen-presenting cells are usually monocyte-derived dendritic cells. So in this case study that I will present today, so we have 10 donors. So we usually take a large number of donors since the MHC2 complex. I think there are like 7,300 different alleles. So every donor, they will react or present different peptides. But usually when you have 10 donors and you can carefully select them to match your bag or your target population, then already with 10 donors, you can actually cover 95% of the MHC2 diversity in your population. So for these 10 donors, we had 4 test conditions. You have 1 negative control, which means that we don't load the antigen presenting cells with anything, and we have 1 positive control, which is the birch major pollen allergen BATV1, which is a strongly immunogenic protein, as some of you may know. So after, of course, we loaded all the cells, then we can, like Professor Purcell already showed, then we can actually purify the MHC2 complexes from the cell surface of the antigen presenting cells. So this is just a schematic workflow. So in this case study, the pellets, they were provided by immune experts, which is a company that actually is specialized in finding the suitable donors and treating them with the test articles. Then we use a lysis buffer to lyse the pellet, and after that we pre-clear the lysate with protein A, and then we can actually run them over a column where the antibodies against MHC2, so the P, the Q, and the R, they are bound to the protein A column, so we can purify the MHC2 complexes. After that, we can separate, after we purify the MHC2 complexes, we elute the peptides from the complex, and then we separate some parts of and then we separate the peptides from other proteins in the complex, and then we can actually just directly identify these peptides with LC-MS. So we have an Evosep, and the Evosep is coupled to the timsTOF Pro, and we do the data analysis, the standard data analysis on a PEAKS server. So of course, the traditional workflow is quite a lot of work. So I think one good lab technician can do like 4 of these manually. But of course, if you want to do MAPS analyses to check the immunogenicity of some test articles, since you need 10 donors and you want to check multiple test targets and also variants of the same test article. Of course, that's not possible to do manually, and that's why we just also tried to make a high throughput protocol, 96-well format high throughput protocol for the purification of the peptides. Of course, Evosep also works in a 96-well format, so which means that our high throughput protocol is actually seamlessly fitting with the LC of the Evosep. We used the 30 samples 30 samples a day method with the performance column and the TIMS, the high throughput or the very fast or reasonably fast 44 minutes gradients of the Evosep. Of course, the TIMS of SCP, which is quite fast and which can take 120 MSMS spectra a minute. So it's a very nice combination. So the combination of high throughput sample processing and of course also the LCMS, the Evosep timsTOF combination is really what allows us to do actually immunopeptidomics fast and in depth. So what looks a typical result? How does it look like? And of course, what is a good result? So we have here the 10 donors. We started off 1 million dendritic cells as a starting material. On average, we have around 8,500 peptides identified. As you can see, there was a spread from 6,000 to almost 12,000. But when you look at the donors, look at the case donor by donor, then you can see that actually the variation within donor is very reasonable. So it's quite good. We get like a robust signal for each donor. So it is expected that donors, of course, that they will give different peptide yields, since of course MHC2 expression can be different, also the antigen processing can be different, and of course also the number of cells in the pellet that we get. So are these peptides MHC2 associated? So one quick check is of course the length distribution. So MHC2, it has slightly bigger peptides than the MHC class 1. So here we see that peptides have a length on average between 12 and 22, and they have a peak around 15 amino acids. So even though we used high throughput methods to purify the peptide, you can see that we see a single peak and that contamination is minimal. So another characteristic of MHC2 bound peptides is that every MHC allele binds peptides with a certain motif, and it, of course, the peptides need to fit the binding pockets of the MHC2 groove. If the allele is known, if it has a known binding preference, then you can predict if the peptide will bind. Of course, the downside is that you have to know the binding motif of the alleles. So that's not always the case for MHC2, especially for DP and DQ. They're not well characterized. But of course, if you have you can cluster the peptides based on the motif, then you don't need that information. So you can see that besides the right length distribution, that first of all, that within one donor, that the results for one donor show the same peptide motifs, and that also different donors have different motifs, as you would expect, of course. So the length distribution and the motifs, they're quite high level, but of course we can also have a look at certain proteins in depth, some control proteins, and we usually look at selected proteins like the gamma chain of HLA2, also called CD74, and also annexin. So what you would expect is that if you have one donor and you have different conditions, that of course that you will detect the same number of peptides, but also if everything goes well, that you get the same peptides for those proteins in the different conditions. And we can see that it's indeed the case for annexin 2 and also for CD74. So this means in general that control proteins, that we get a robust and reproducible signal, so indicating that the match protocol is reliable. the total number of peptides, it's quite high, but that's also necessary since, of course, if you add a test article to an antigen presenting cell, you will actually have a lot of self peptides, and just to find the non-self peptides, that is very tricky, because of course you need to find these small amounts of peptides. So it's critical that we get a decent amount of these peptides, and generally is said that around 1,000 to 5,000 peptides is actually good to get a good map signal, but you can see, of course, we can identify substantially more, and also we can actually see that for our positive control, the BATF1 peptide, we are on average, we can find 40 peptides in the sample. So this is good, and we can find a strong signal, but of course, BATF1 is strongly immunogenic. But on the other hand, BATF1 is also a very short protein, so 40 peptides for BATF1 is certainly a very sensitive measurement. When you look more into detail in separate donors, and you can see that these peptides, they are clustered into regions, and the immunogenicity of a region is usually defined or said the region is immunogenic if you have a series of peptides that are mapped onto that region, so a cluster. So what we also can see is that different donors have different immunogenic regions. As we can see here for donor 1, the immunogenic regions are substantially different than from donor 2. So in the end, we can conclude that our positive control proves that the MAPs will identify immunogenic regions sensitively. Besides that, we are also interested, of course, how specific our protocol is. So the peptides matching to BATF1, they were luckily only identified in BATF1, so there's no cross-contamination of the samples. So peptides matching the test articles were also identified only in the corresponding condition. So here's a heat map, so where you can see that the peptides, the proteins that we need to find, are only found in their corresponding samples. So of course, we are quite confident that high throughput purification works well, but to get high quality immunogenicity data, of course, we also need a very robust LC-MS setup, and the Evosep 1 with the performance column and the 30 samples a day method gives us exactly that. So in total, we had 60 MAPS analyses. Between every sample, we do have like a QC standard, in this case, the glufit peptide. So but this means that we have like 120 runs in this protocol or in this case study, and that we can already do that in 4 days of instrument time. So it's quite fast compared to a few years ago. And also importantly, no carryover is observed in the Evosep system. So if we look at just the quality control of the retention time, then you can see that during the whole project, the whole case that we were measured, the retention time was very reproducible, as is also the peak width. So immunopeptidomic samples can be challenging, but we see that the Evosep is able to, on long term, to give very high quality separations. And also coupled with the timsTOF Pro, the timsTOF SCP, excuse me, we can get actually very reproducible LC-MS measurements. So to summarize, so we showed that the sensitivity and specificity of our maps is actually at a level that allows us to do like in-depth immunogenicity studies, and we can actually help with de-risking the various biotherapeutics. So we measured 60 samples, and in total we identified 90,000 peptides in the total experiments, which I think is quite a substantial number of peptides. And also, if we look at the samples from the same donors, so we get always roughly the same numbers of total peptides identified. We get the same motifs and distributions. And we can also see with the QC proteins that the signal that we get is always very robust. So in the end, we can say that at the moment we have a MAPS assay that the Evosep 1 allows us to actually get most out of high throughput sample preparation with its perfect balance between speed, robustness, and high sensitivity. So that's needed for a nice assay. So, and then of course, I want to thank my co-founders, Geert and Lies. And if you have any questions, I'll be happy to answer.